Thank you for the invite to be here today. So my big question for today is, can we chip away at disease, focus being on chip? And what I'll be talking about today is a really rapidly growing technology called organ on a chip. And some of you may have heard of this before, you may know little bits, but hopefully by the end of today you'll know a lot more. So I'm going to start my big question by breaking it up into lots of smaller questions. So organ on a chip, why, what, how, and who? So why do we need organ on a chip? What is it, and what do we use it for? How do we actually build an organ on a chip model? Who's working on organ on a chip, and why is it exciting? And hopefully you all agree it is very exciting by the end of this talk. So a little bit of biology to start with, and apologies if this is really simple, but it's quite foundational for my talk. So when we look at organs, so we've got an example here of the lung. These organs have a hierarchy to their structure. So the organ is made up of tissue, which is made up of these functional tissue units. So these units, collections of cells that actually give the organ its function. And then these cells are the building blocks of these units. So for one example here of the lung, which we'll come back to. Another example here of the kidney, exactly the same. So we have the organ, which is formed of these nephrons. The functional unit of the nephron is the corpuscle, and then this is made up of the kidney cells. We'll come back to this and why it's important a bit later on. So that's brilliant. We understand the structure of the organs, but what's interesting to researchers often is what happens when these organs go wrong. And in order to fight diseases that may occur in these organs, we really need to understand how they start, understand how they progress, develop new treatments, and then also to test these treatments. And the way that we do this is using models. So I want you to all think about what I might mean by the term model and sort of have a picture in your head as we go on to the next slide and we start talking about models. So some of you may be thinking of runway models, and I'm sad to say that's not what we're talking about today. Some of you may be thinking of sort of miniaturized vehicle models, so, and maybe it's a bit dated I suppose, but Airfix or Hot Wheels, so sort of miniaturized versions of a more complex, larger structure. And some of you may be thinking about computer models, or even mathematical models. And what these all have in common, sadly apart from Naomi Campbell, is they're really just simplified representations of a complex system or process. That's what we mean by the term model. So I'm a biologist. What does this mean in biological terms? So again, it's really just sticking the word biological in there, and it's the same sort of definition. So a biological model is just a simplified representation of a complex biological system or process. And what this means in real terms is that we can develop these models that are just a simplified representation of something more complex. The problem is that the options that we have for modeling in biology currently are quite limited. So we either have what we call in vitro models, so cell-based models where we culture cells on plastic in the lab, and these will sit in an incubator. But I think you'll agree that none of us look like this. It's not particularly physiologically representative. It doesn't really represent how our body functions and even how our cells function. So another option that we have where we can add in a bit more complexity is in vivo or animal models. But again, I'm not going to offend anyone, but no one in this room looks like this. And again, we just don't have that physiological relevance. Our bodies behave in a very different way to that of a mouse. So what we really need is better models, and I'm hopefully going to convince you today that organ on a chip is the answer to that. So hopefully that's addressed part of our first question, why do we need organ on a chip? And now, I haven't even told you what it is yet, so we'll get into the interesting bit now. So, organ on a chip is really, really simple. It's just a combination of biology and engineering that allows us to build better models. That's it. So it's a very weird name for it. Organ on a chip sounds really weird, but all it is biology and engineering coming together to give us better models. And this basically, I mean, there's some examples here of what these organ on chips look like. I've got one here in my hand so you can sort of see the scale of them. So tiny, tiny little devices. But all it is cells that we showed at the start, so the building blocks of organs, grown in these really small chambers and channels. They're provided with everything that they need to survive and behave as they would in the human body. And we have really precise control over their internal and external environment. So again, we're trying to improve on these super basic cell models while also moving away from animal models because we can use human cells in these organ chips. So we're developing these improved models. So just to give a bit of an analogy of how this works, what it actually is, I'm going to use the analogy of growing plants in a greenhouse. So you'll have to just bear with me. I know the scale's a bit off for this compared to a greenhouse, so imagine everything shrunk right down. But what do we need to grow plants in a greenhouse? So we need some sort of support material, so in this case soil to grow the plants in. We need seeds, and not just any seeds. If we want to grow tomato plants, we need tomato seeds. If we want to grow, I don't know what they are, beans or something, then we need bean cells. So we can't just chuck any old seed in. We need a seed that's going to grow the plant that we're interested in. We then need nutrients, so in this case fertilizer to help the plants grow. We need to control environmental factors, so using the greenhouse we can control temperature. We can also control the carbon dioxide, oxygen, the gases coming in and out. And then finally, we need to remove any dead plants that might affect the culture. You'll have to bear with me on this, but if we swap the greenhouse for an organ chip, and I'm going to try and convince you now that this is a perfect analogy. So the support material, rather than being soil, we have something called a matrix, and this is effectively just proteins that support the cells. The cells sit within these matrix, and it will just support the growth and the function of the cell. So matrix is the support material. The seed is the cells that we put in. So again, just like growing a tomato plant, you need tomato seeds. You need the right cell for the organ that you're trying to mimic. So if we go back to the lung and the kidney at the start, if we want to mimic a lung, then we need to put lung cells in. If we want to mimic a kidney, then we need to put kidney cells in. The same as growing a plant, you use the cells that are from that particular organ. Nutrients, so we don't want to put fertilizer in these chips because they will get infected and the cells will die. So we have something called media, which again is just a term for, it's basically just a soup that the cells love. It's a collection of proteins and carbohydrates that keep the cells happy. So that's the nutrients that go in. We need to control the environmental factors in pretty much the same way, so temperature and gases, but this is all done by putting these chips, obviously you can see they're very small, they'll fit into an incubator, and that can all be controlled in that system. Waste removal is also important, but in this case I've called it waste, because it's waste for the cells, but for us it's really important. We collect it, we can run analysis on the waste media, and that will tell us a lot about what the cells are doing in the chip, so it's not really waste for us, but it is for the cells. The thing with organs, which will set them apart from the plant analogy, and this is where the analogy possibly falls apart a little bit, is that organs aren't static. You know, many of the organs in our body experience mechanical forces. So whether that's peristalsis in the gut, the heart beating, the lungs expanding and contracting as we breathe, or the one that I'm particularly interested in is joints. As you move around, your joints will move. So it's really crucial that we can also apply mechanical forces in these organ chips, which we are able to do. So this is where we start to come away from the greenhouse a little bit. And many of these organ chip systems, and I'll show you later, there's lots of them, but we're able to apply mechanical stimulation. So again, really mimicking far more closely what's happening in the body than we would be able to with traditional models. And then the other thing is it's brilliant to be able to grow a perfectly healthy tomato or a perfectly healthy lung in one of these chips. But what we're really interested in is modeling diseases. So we need to be able to generate disease models, and many diseases have factors that are released by the organs and act on other organs, which will then cause the diseases, which we just call biochemical factors, and we're also able to add those into these models. So we can develop a healthy tissue, add in biochemical stimulation, and turn it into a diseased organ on a chip. So we can study diseases as well, and the way that they process. Okay, so back to this, the picture from the start. And I think what's really important to understand here, and this is something that came up at the Youth Forum, so thank you very much if anyone was at the Youth Forum, that's really useful to talk about. So this is called an organ on a chip, but we're not trying to replicate an entire organ. We're not even particularly trying to replicate an entire tissue. What we are trying to replicate is the functional unit of these organs. So we're mimicking function by using the building blocks of these functional units in these chips to mimic the function of the whole organ. But I think you'll agree this looks nothing like a lung or a kidney. It's the function that we're trying to replicate. So just to make that really clear. Okay, so hopefully we've addressed what is an organ chip and what we use it for. So now we're going to talk about how we actually build an organ chip. And I thought the best way to do this would be for us to sort of build one together in the room today. So as I said, and quite selfishly, I'm going to talk about what I'm really interested in, which is the musculoskeletal system, particularly the human knee. So this is all of my research is focused on the knee. And this is often how we start to build an organ on a chip. We find an organ that we're particularly interested in, and then we start to build around it. So the knee, and this is something that's come up before as well, but we do consider the knee as an organ, which I think people do find quite strange. You know, if you think about organs, you think about lung, liver, heart. But I will try and convince you that the knee is also an organ. It's multiple tissues working together to to provide a particular function, which is really the definition of an organ. So we do treat the knee as an organ in both health and disease states. And it consists of multiple tissues. So there's the synovium, which is sort of a membrane that encases the joint, the meniscus, which is a bit of a shock absorber on the sides, the cartilage, which provides friction, low friction movement as you walk around, and then the bone, which is the main supporting structure. So we've got our organ of interest, but what we're really interested in is what happens when this organ goes wrong. And another way that we start to build these models up is by thinking about a disease. And in this case, I'm particularly interested in arthritis. So I'm sure you've all heard of arthritis. It's the most common joint disease in the world. But the problem is we don't really know how it starts or how it progresses. We don't really know how we go from this lovely healthy knee to this arthritic knee. But we are pretty sure, and the evidence is building, that two tissues that are involved are the cartilage and synovium. So these will be the focus for our organ chip, which we'll build here today. The next decision we need to make is which chip to use. As I said before, there's lots of different chips on the market now, and you really need to pick a chip that's going to help you address your question. In this case, we're using the Emulate chip S1. So this particular chip consists of two channels. So this is a cross-section through the center. So a larger top channel, and then a smaller bottom channel. And then these two channels are separated by this flexible porous membrane. So this means that cells in the two channels can communicate with each other without coming into contact with each other. So as I talked about before, the factors that the cells released can pass through and so the cells can talk to each other. And what people tend to do is seed the organ-specific cell in the top and then a blood vessel cell in the bottom. So what you're mimicking is sort of a vascularized tissue. The beauty of the Emulate chip is that these are vacuum channels on the side. So when you apply a vacuum to those, it will stretch the chip and therefore apply mechanics. So we use that to mimic walking in this particular instance. So that's our chip selection. I'll skip that on. So we've looked at the organ of interest. We've looked at the disease that we're trying to unpick. We have our chip ready. So let's start and build the model as we did before. So the support material, the matrix that I talked about before, and in this case, we're using two different matrices or matrices. One for the cells from the cartilage and one for the cells from the synovium. We then isolate the cells. So we take cells from the synovium, which we call fibroblast-like synoviocytes, and we take cells from the cartilage, which we call chondrocytes, and these go into the chip in the two different channels. I talked about nutrients before, and we used the media that we had before, and this media is specifically designed to keep both of these cell types happy, which is more of a nightmare than it sounds, but it works. Environmental factors, as I said before, we control temperature, we control gases by putting these chips into the incubator. The mechanical stimulation in this case, as I said, we can apply a vacuum to these channels here, and this gives us stretching across the membrane. So those cells are experiencing stretch, and it's cyclic stretch, so to really mimic walking. And we can change the frequency of this to mimic faster or slower walking. Waste removal, again, it's not waste for us. We do a lot of analysis with this. And then critically, one of the ways that we think arthritis starts in the knee joint is through inflammation. So we can add in inflammatory factors here to try and mimic the onset of the disease. So that's the model that we've built together. This is the result of it. So again, I'm showing the cross-section here with the larger top channel, smaller bottom channel. We have the cells from the synovium here sat on their matrix, and then the cells from the cartilage here sat within their matrix. And if you look to the right-hand side here, this is what we call a live dead stain. So live cells show up green, and dead cells, there are a couple. There's one there and one there. They'll show up red. So this is showing the cells in the top channel, and they all look pretty happy and healthy. Hopefully you'll agree. And then the cells in the bottom channel. And you might notice that these cells are very rounded, very punctate. And that's because they're completely suspended in the matrix that we talked about. So they're held in 3D, which this particular cell type really likes. I do have a video here which shows it slightly better. So if I pause it here, then you should be able to see that the cells in the bottom channel, so the chondrocytes, are spread out in 3 dimensions because they're suspended in a gel. And then we have this nice monolayer of the synoviocytes, the cells from the synovium in the top channel. So that's the model, looking really at the interaction between cartilage and synovium. But what do we actually use it for? It's brilliant, developing these models, but we have to use them for something useful, otherwise they're pointless. So this is, again, I'm being very selfish talking about my work, but this is my current project. So we're really using this model to address clinical questions. And as I said before, the beauty of these systems is that we can use human cells in the chips. So what I do is speak to surgeons, and waste tissue coming from surgery, so patients undergoing knee replacement, for example, will come to me in the lab, I can extract cells from there and then develop these models. And what we're really interested in is developing what we call stratified or personalized models. So stratification is where we group patients into certain groups, and then personalization is more individual. So we're using this to develop stratified models of different disease states, different disease stages, so early versus late arthritis, and also personalization. So there's some patients that don't respond very well to particular treatments, and we want to try and use this to find out why by developing personalized models for these patients. So just a bit of a flavor of what we're using it for. So hopefully that makes sense of how we build the organ-on-a-chip, the questions we ask ourselves, and why everything goes into them. So now I'm going to talk a bit about who's interested in these. So lots of people is the short answer. So we have engineers who are actually developing the models, coming up with the materials that work, the systems that can apply mechanical stretching, things like that. Clinicians, so as I said, I'm in constant contact with clinicians who provide tissues to go in the chips, and they're interested in how that can then help their patients further down the line, if we can stratify or personalize. Pharmaceutical companies, so this is one of the big interest groups. So many drugs are still tested on animals in the current environment, and while that's bad for the animals themselves, it's also not very good science. As I said before, animals are very, very different to us in terms of physiology, and so 90% of drugs going through clinical trials actually fail after the animal testing stage, so it's a massive waste of money, waste of animal lives. So pharmaceutical companies are really interested in this technology to go somewhere in that pipeline to improve their outcomes and save them money, basically. Biologists like me, because we can build better in vitro models that help us to understand diseases and come up with new ways to treat them. And policymakers. So we've had quite a lot of interaction with government recently. They're interested in driving this work as well. There was a big policy change in the US last year, or 2022, where drugs no longer have to be tested on animals. So if something similar were to happen in the UK, this sort of technology is something that we'd be looking at to try and drive that replacement. And the other groups that are really interested are charities, so particularly charities that look at animal welfare, really interested in this sort of work. So my project last year was completely funded by the National Centre for Replacement, Refinement and Reduction of Animals, and the idea being that if we can make these models good enough, then other scientists will use them instead of using animals in their research, which is really nice and drives better science. And in terms of who else is interested, I said before there's lots of different types of chips now. When I first started, there were very few companies, and it's really taken off in the last few years. This is just a snapshot of some of the companies that are currently making organ chips. The ones that we currently work with are Emulate, which is the one that I've been talking about the whole time, CM Bio, Mimitas, Tissues, I've got CM Bio twice, and Biomimics. And they all have a different setup, different purposes. So it really is a case of picking the best chip to answer your question. And hopefully we've covered quite a lot of them now. So finally, and hopefully you'll agree that it is exciting now, but why is it particularly exciting? So firstly, the technology itself, these are living, breathing models that we can use in the lab to answer lots of different questions, which really sets them apart from any of the other types of models, apart from Naomi Campbell, that I showed at the start. So really, really exciting stuff. And then it's the potential, so the applications of what we can actually use these models for. So as I said, as a biologist, this is probably the most interesting one for me. We get to understand how tissues develop, how they function normally, and ultimately how that goes and diseases form. So it gives us a really, really good chance to understand tissues and diseases. We can use them for testing treatments. So as I said before, they're really good for drug screening. This is why a lot of the pharmaceutical companies are really interested. And for all of these different facets, we can drive animal replacement in research, which is really, really important, not just for animal welfare, but for science and the robustness of the science. And crucially, we can use this for personalized medicine as well. So by putting individual patient cells into these chips, we can understand the biology of their tissues, understand how their disease in particular is progressing, and also test personalized treatments. So I think it's really exciting, and hopefully you agree. So we've addressed all these smaller questions. The only thing left to do is to address the big question, which is, can we chip away at disease? And hopefully you'll agree with this answer. You may have to squint to read it. It looked better on my laptop screen. But I believe, and hopefully you will agree in some way now, that yes, we can use these, and we will be able to chip away at disease over the next few years. So thank you.